but that is one of the primary functions. The respiratory system is involved with bringing those gases to the circulatory system. In many books, in many references, you'll sometimes see them linked together, referred to as the pulmonary circulatory system. So we're thinking of it as one system that's involved in bringing gases and then transporting those around the body. Our book separates them into two. Now, when we talk about the circulatory system, we're talking about blood. Blood is a special connective tissue. If you recall, when we're talking about the tissue types, there's regular connective tissue and special connective tissue. And the special connective tissue is recognized by having an extracellular matrix. With blood, the extracellular matrix is a fluid that is referred to as plasma. And in fact, approximately slightly more than 50% of blood is made up of plasma. There are things dissolved in the plasma. There are functions that the plasma has, but that is comprised of the blood. The remainder of the blood, a little bit less than 40%, is what we refer to as formed elements. These would include things like the red blood cells, the white blood cells, and the platelets. Now, the functions of blood can basically be broken down into three categories. The first of these is transportation, moving things around. What does blood move around? Nutrients that it picks up from the... Where do you get your nutrients? Digestive system. What else does it move around? Oxygen and gases that it picks up from the cells if it's carbon dioxide, or from the lungs if it's oxygen. It's also involved in moving hormones around. It's also involved in moving regulatory proteins around and so forth. So it's really a sort of the network of transportation. It's the highway within your body where everything can travel around to any part of the body. Every single cell in your body is usually within a few microns, a few micrometers away from a capillary. So anything that can be produced in one cell can be transported to any other cell in the body. It's also involved in regulation. We talked a little bit about how you have sensors that can detect things like blood pressure. They can detect things like glucose level, the pH of the blood, and these are all involved in regulatory mechanisms. So blood is a very essential system for helping to regulate the body. And then lastly, it's involved in protection. How is blood involved in protection?
Clotting.
Clotting and white blood cells. So the blood is involved in an immune response. If there's a pathogen that invades, the white blood cells are involved in trying to fight that pathogen. It's part of that system. But it's also involved in a physical protection in the clotting system, where if you have a leak, if you have a break in the blood vessels, that will help heal that wound. So if we look at the blood itself, as I mentioned, slightly more than 50% of the blood, right around 55%, is comprised of plasma. Plasma is this extracellular matrix that's produced by the red blood cells, and it's a fluid that would help sort of aid this process of transportation. There are a lot of things that are dissolved in plasma. There are proteins. Plasma primarily is made up of water. There are solutes that are dissolved in there as well. The remainder of the blood, the formed elements, comprises about 45%. Most of this is red blood cells. So they're the ones that are moving the gases around, so we need to be able to transport gases from and to and from cells. So that is going to be the majority of the remainder of the blood cells. However, we do have some platelets. These are involved in clotting. And we do have some white blood cells, multiple types that are involved in the immune response and body protection. So blood plasma, as I said, is about 92% water. In addition to that, we also have a variety of solutes. There are going to be wastes that are dissolved in plasma. There are nutrients that are dissolved in plasma. Hormones are dissolved in plasma. So when the body produces a hormone and sends it around, that's going to be in the plasma within the bloodstream. There are various ions and a lot of different kinds of proteins that are dissolved in plasma, including albumin, fibrinogen. And if you remove all of these elements, if you remove these proteins, the nutrients, the ions, you no longer have what we refer to as plasma. Instead, you refer to it as serum. And serum can be used for health purposes. It can be used to test to see if someone's been exposed to a disease and so forth. You can actually donate serum and so forth when people need additional plasma. Sometimes when you need more blood, you actually just need a volume, and so they can donate plasma or serum. So the remainder of that, the 45%, are these formed elements, primarily comprised of red blood cells, which are referred to as erythrocytes. There are about 5 million erythrocytes per microliter of blood. That's a lot. They're incredibly critical. One thing that's unique about mammalian erythrocytes is that they don't have a nucleus. So here we're talking about a cell that doesn't have a nucleus. Basically, once the red blood cell is fully formed, it is no longer considered alive. It's just involved in this transport process. Now, the red cells of the vertebrates all contain hemoglobin. What does hemoglobin do? It transports oxygen. What does the oxygen molecule bind to? Iron. And so the hemoglobin contains this iron that helps move these oxygen around. Do the blood cells of non-vertebrates contain hemoglobin? Yes, no? Yes. Some of them do. There are actually some insects that have hemoglobin in their blood. It looks red, and whenever you see bloodworms in water, little tiny worm-like things that are bright red usually, they actually have hemoglobin in their blood, just like vertebrates. But not all invertebrates do. Vertebrates, the red blood cells have hemoglobin. Some of the invertebrate blood cells do. So the other formed elements include things like the white blood cells. These are referred to as leukocytes. This is comprised of less than 1% of all the blood cells, so they're relatively rare. They move around through the body, and they can actually control where they're flowing, where the red blood cells are basically just flowing in the direction that the blood is moving. So the white blood cells are involved in body defense. These are things that can recognize invading pathogens, invading viruses and diseases. They can then actively fight those diseases. They will send out signals to the body if there's a wound, if there's a pathogen, if there's some sort of immune response that is needed. They're larger than erythrocytes, and they are alive. They do have a nucleus. So they are basically, you can almost think of them as police patrolling the bloodstream, looking for these invaders. They can actually migrate in and out of the blood cells as needed. They can or in and out of the circulatory system as needed. So if there's a pathogen in your skin tissue or something like that, they can migrate out and start attacking the pathogen there. They don't need to wait until the pathogen invades the bloodstream. White blood cells, the leukocytes, are broken down into two types. You have the granular leukocytes. These include the neutrophils, the eosinophils, and basophils. You also have the agranular leukocytes. These are the monocytes and lymphocytes. They're involved in different stages of pathogen resistance, different stages of fighting off diseases, bacteria, and so forth. And as you can see already, we have the circulatory system that's now tied to two different systems. We have the respiratory system that it's tied to, but now we have the immune response system where the circulatory system plays a role as well. The last thing in the formed elements are the platelets. These are basically fragments of cells that break off during the process of producing larger blood cells. Large blood cells are produced in bone marrow. In that process, you sometimes have these fragments that break off. These stay in the bloodstream. They're called platelets, and they're involved in the formation of clots. Why are clots important?
So that you don't bleed out.
So you don't bleed out. Every time you have a hole in your circulatory system, you're losing blood. Now, if you have a tiny little prick on your finger or something, and you have a little tiny drop of blood, it's not that much. It's not something to worry about. But if you start getting slightly larger cuts, bleeds a little bit more, you need to have some way to sort of plug up that hole. That's where platelets play a role. Platelets are floating around in the bloodstream just like the blood cells. If the vessel is damaged, here we have someone sticking a needle into their vessel, maybe to draw blood or something. The platelets, as they're flowing across, they sort of get exposed to this, and they sort of become a little bit sticky and adhere to the capillary lining where this damage has taken place. They start forming this plug to stop the blood flow out. There's a cascade reaction where you take a different series of enzymes and ultimately produce fibrin, which is used to produce clots. Those clots also then adhere blood cells. They adhere white blood cells, red blood cells. We need the white blood cells there because in case there's any sort of pathogens, any bacteria or something to fight where this entry point into the circulatory system is. Eventually that clot will completely cover over this wound, and the wound can then heal. So the platelets are very critical. There's certain genetic disorders where people don't have enough platelets. They aren't able to produce clots. They have to be very careful if they're wounded because even the smallest wound can produce a situation where they're going to bleed out. Now, circulatory systems are broken down based on the kind of animals we're talking about. With invertebrates, we see a wide variety of circulatory systems depending on the invertebrate itself. It has to do with the size of the invertebrate, it has to do with the complexity of the invertebrate, and it has to do with the lifestyle of the invertebrate. Some invertebrates don't have a circulatory system at all. They're still animals, but maybe they're small enough, they don't need this sort of system of transporting materials around. Sponges and cnidarians primarily use water from the outside environment as their circulatory system. So it's just this process of diffusion within these organisms that allows things to move around. They don't need an actual transport system. Nematodes have a body cavity, but they still don't have a circulatory system. There's fluid in that body cavity. They basically use that for circulation. There's a small flow of that fluid, so material can dissolve into that fluid and then be transported throughout the entire organism. But these are all very small organisms. When you start to get larger, you need to have some sort of mechanism for efficiently moving things around. The larger you are, the less efficient diffusion becomes. So larger animals, larger invertebrates need to have some sort of development of a circulatory system. It's going to be involved in transporting nutrients around. It's going to be involved in removal of waste. It's going to be involved in moving gases around. Now, circulatory systems in large animals are categorized as either open circulatory systems or closed circulatory systems. This basically has to do with what's actually going on with the vessels themselves. In an open circulatory system, there's no distinction between fluid that is in the circulatory system and fluid that is out of the circulatory system. There are some vessels, there are some tubes that help sort of direct the flow. There is a heart that helps create a circulation. But the tubes open up into the hemolymph, into the body cavity. And so there's no distinction between fluid that is in the circulatory system and out of the circulatory system. So the fluid in this is all referred to as hemolymph. Now, a closed circulatory system in invertebrates is going to be overall similar to what we see in vertebrates. There's going to be a distinct circulatory fluid that is moving around a closed network. There may be diffusion in and out of it, but it's going to be separated from the extracellular fluid. So with a closed circulatory system, you have a separation of extracellular fluid and circulatory fluid. So this is sort of what we see in invertebrates. There are many invertebrates that do not have any circulatory systems whatsoever. They just rely on passive flow or diffusion to move things around. Some invertebrates have an open circulatory system. They do have hearts, they do have some vessels, but the vessels open up into the body cavity, and so they move this hemolymph, and the whole system, all of the fluid, is being moved around through the circulation process. And then lastly, we do have some invertebrates that are similar to vertebrates in that they have a closed system. You have a heart, you have fluids, you have a movement of the fluid in a unidirectional flow. You have smaller fluid, but it's a closed system. You have a separation of the fluid that's in the circulatory system and the fluid that's out of the circulatory system. Now in vertebrates, all vertebrates have a closed circulatory system. In vertebrates, what we see is we see an increase in complexity of the circulatory system related to the evolutionary age of the invertebrates. The oldest vertebrates are fish. They have the simplest circulatory system. They have what we refer to as a true two-chambered pumping heart, meaning the heart in the fish have two different chambers that are involved in moving the blood around. The heart itself is sort of comprised of four different structures. They're arranged in a linear fashion to help increase the efficiency at which fluid can be pumped around. In the first chamber of the two-chambered heart, you have two structures. You have the sinus venosus and the atrium. Blood from the body will flow into these, and then they will get pumped into the second chamber. The second chamber also contains two structures. You have the ventricle and the conus arteriosus. So blood from the first chamber gets pumped into the second chamber, and from there it gets sent out. So again, you have a directional flow. It allows for more efficient processing. Now, when the fish heart contracts, basically each of these four structures will contract in that order. First the sinus venosus, then the atrium, then the ventricle, then the conus arteriosus. So it's just sort of a linear process of this contraction. Once blood leaves the heart, it then travels to the gills. What is it doing at the gills? It's picking up oxygen. From the gills, it then travels through the rest of the body where that oxygen is sent along to the cells. Carbon dioxide is picked up. It then moves back to the heart. So what you end up seeing is this single circuit, a single loop in the fish circulatory system. It starts off at the sinus venosus, moves to the atrium, moves to the ventricle, into the conus arteriosus. Then it moves to the gills where it becomes oxygenated. Does it magically turn red? No. Then moves through the body. The oxygen is removed from the blood by the cells. Eventually, the deoxygenated blood goes back to the heart, where it goes back to the sinus venosus. So that is the process where you have the blood moving through this single loop, this single circuit. So the fish circulatory system is referred to as a single circuit. Amphibians, we start to see a higher level of complexity. We have an evolution of a lung. The lung is a unique structure on terrestrial vertebrates. This coincided with the evolution of a second circulatory loop, a second circuit. So in amphibians, we start to see what we refer to as double circulation. There are now two loops that the blood flows through. So the double circulation, this double circuit sort of divides the blood into blood that is moving between the heart and lungs and blood that is moving between the heart and the body. Some of the blood will move from the heart to the lungs, pick up oxygen, then move back to the heart. That's one circuit. Some of the blood will move from the heart to the body where the oxygen is removed, goes back to the heart. That's the second circuit. So you have two sort of loops in this amphibian circulation. The pulmonary circulation refers to the blood that is moving between the heart and the lungs. So anytime you see the word pulmonary, you should be thinking heart and lungs, oxygenating the blood. The systemic circulation is the circuit that involves the blood moving between the heart and the body. So you sort of have different names now for these different circuits. Pulmonary circulation, heart and lungs. Systemic circulation, heart and body. As we see an increase in complexity of the circulatory system, we also see an increase in complexity of the heart. So the amphibian heart is now a three-chambered heart. The fish heart was two chambers. We see an evolution of a third chamber in the amphibians. There are two atria now. There's still only one ventricle. One atria receives the blood from the systemic circulatory circuit. One atria receives blood from the pulmonary circuit. But they both move that blood into this single ventricle. So you have a partial separation of these two circuits. They're still sort of together. The blood that enters the ventricle, some of it is from the pulmonary circuit, some of it is from the systemic circuit. It ends up getting mixed together in this ventricle. So it's an incomplete separation. It's not as efficient as if they were completely separated. Because when blood leaves the ventricle, some of it will go into the pulmonary circuit, some of it will go into the systemic circuit. So it splits into the two circuits after it leaves the ventricle. Now, amphibians are able to do this because they do also get oxygen from their cutaneous respiration. They are able to have this somewhat inefficient mechanism because they have another mechanism of getting oxygen. In reptiles, we see a more advanced system. They also have a three-chambered heart. They also have the separation of the circuits. But in the heart, we have the evolution of a septum that partially divides the ventricle. It doesn't divide it into two chambers. Then we'd have a four-chambered heart, not a three-chambered heart. But it partially divides it to increase the efficiency. There's less mixing of the blood from the two circuits. Why do you care about this? Well, because the pulmonary circuit is where the blood is picking up oxygen. It's a waste to have that blood go back to the lungs. The systemic circuit is where blood is being deoxygenated. It needs oxygen. It's a waste to have that blood go back to the systemic circuit. So by increasing the separation of the circuits, you increase the efficiency of the process. So it's a more efficient system than we see in the three-chambered heart of amphibians. So this is what we see in a three-chambered heart. There's two atria. One of them is involved in receiving deoxygenated blood. One of them is involved in receiving oxygenated blood. They both pump into a single ventricle. Blood that leaves the ventricle either goes to the systemic system, where it's going to be moved by oxygen is removed by the cells, or it goes to the pulmonary system in blue, where it's going to be where the blood is going to pick up oxygen from the lungs. Overall, this is what we see with the dual circuit now. We have one circuit that's a pulmonary circuit. Deoxygenated blood leaves the heart, goes to the lungs, picks up oxygen, and goes back to the heart. We have oxygenated blood leaving the heart in the systemic circuit, going to the body, picking up where the blood is, or the oxygen is used, then it goes back to the heart. But because you don't have a complete separation in the ventricle, some of this blood is going to be mixed. You might have oxygenated blood going back to the heart. You might have deoxygenated blood going back to the body. With reptiles, we see a partial division, a septum form that partially divides the ventricles. This increases the efficiency, decreases the ability of that blood to mix together, but it still is a three-chambered heart. It's not until we get to mammals, birds, and crocodiles that we see the evolution of a four-chambered heart. A four-chambered heart now completely separates those two circuits. The blood that is going in the pulmonary circuit, when it is not going to mix with the blood that is going to the systemic circuit. So you have two separate atria. You also have two separate ventricles. There's a complete separation. Now what happens in these four-chambered hearts is that the right atrium receives deoxygenated blood from the body. So this blood now does not have oxygen. Eventually it's going to need to be sent to the lungs. The blood is delivered from the right atrium to the right ventricle, which then pumps this blood into the lungs, where it can pick up oxygen. The arteries that are involved in this are referred to as pulmonary arteries. They deliver deoxygenated blood from the heart to the lungs. We usually think of arteries as having oxygen, but here we have pulmonary arteries that deal with deoxygenated blood. The pulmonary veins deliver that oxygenated blood that has been picked up, where it's picked up oxygen from the lungs back to the heart. So again, if we see the word pulmonary, we're thinking between the heart and the lungs. The left atria then receives blood from these pulmonary veins. This is now oxygenated blood. It goes from the left atria, moving it to the left ventricle, which then pumps this blood to the rest of the body. This is the systemic system. So the systemic arteries are taking oxygenated blood away from the heart to the rest of the body. The systemic veins now take deoxygenated blood from the body and deliver it to the heart. So if you look at what's going on, we have a complete separation. We have the pulmonary circuit. We have the systemic circuit. We don't have any mixing of the blood that takes place in the heart. This is a more efficient mechanism for moving blood around, for making sure that blood that needs oxygen goes to the lungs, blood that has oxygen goes to the body. When we look at sort of this phylogeny of the evolution of the heart, we see increased complexity as we move up into evolutionary time, but we see that the four-chambered heart has evolved twice. Crocodiles have a four-chambered heart, or rather, crocodiles have a four-chambered heart. Mammals and birds have a four-chambered heart. So there's two distinct periods where this four-chambered heart has evolved. Most reptiles have a three-chambered heart, same as amphibians. Fish have a two-chambered heart. When we deal with invertebrates, we don't have a distinct separation between the chambers at all. On Friday, we'll continue this, talking about the cardiac cycle. If you have any questions, please let